Okay, good morning, Year 10 triple scientists. Welcome back to the Plant 2 topic. Today we are looking at the question, what is the effect of light and gravity on a seedling? This is the second half of the required practical. We looked at the first half last lesson, and this is kind of our results, what we'd expect to see, and also how can we handle the data? What kind of data is it that we're actually going to be collecting? So, driving question at the top, what is the effect of light and gravity on a seedling? This is the required practical part 2. Keywords are the same as last lesson: hormone, auxin, tropism, diffuse, phototropism, and germination. We are going to be trying to explain what we see as well. So the hormone, auxin, and tropism, and diffusion, and phototropism are going to be more useful in today's lesson. Again, and we're going to have a little start looking at the variables. Before we get going, why are we learning about plants? By now, hopefully, you should be able to tell me this. Basically, plants are the reason that all life on Earth has energy, effectively. So all animals require energy that plants are able to capture from the sun. Plants are called producers, and this is because they are setting off the food chain, really, because they are able to start it off with the energy. Also, we rely on plants for crops. Even if you're a meat eater, your meat requires plants too. By understanding plants, we're able to help protect the environment, which we are obviously definitely part of, even if it doesn't feel like it when we live in a city. Right. For the investigation, investigate the effect of light on the growth of newly germinated seedlings. Can you tell me what the variables are? Can you remember these from last lesson? Pause the video, we'll go through them when we get back. Hello? The independent variables for this investigation is the place that the plants are. So in light, dark, and partially light conditions. The dependent variable, this being the thing that we're looking at the effect on this, and that would be the mean height of seedlings. Again, that's the mean height, so that means the average height. So we're going to have to calculate some averages today. And the control variables are the number of seedlings on each dish, how many there are, how spread out they are, the volume of water each seedling is given, and importantly, the temperature that they are kept at. It would be no good to put some on a really hot windowsill and then others in a cold cupboard, because even though you are changing the light, you're also changing the temperature, and that is not the point of this investigation. Again, germination is basically the first growth of a plant out of a seed. So this is leading up to it becoming a seedling. Seedlings have got roots, they have got leaves, they're just little plants basically. Germination is just straight out of a seed. Quick recap on what the investigation is. I'll just go through the method now. You put cotton wool onto 3 petri dishes and add the same volume of water to each dish. Add 10 seeds to each dish and add 10 seeds into each dish and place them in a warm place where they won't be disturbed. This will allow them to germinate. Add more water if the cotton wool dries out. Okay, so you've now got 3 trays of germinated seedlings. Once the seeds have germinated, ensure the petri dishes each contain the same number of seeds. If any of them haven't germinated or anything, remove them. Make sure that they've all got the same number. One petri dish will sit in full light on a windowsill. The second will be placed in a dark cupboard, and the final dish will be placed in partial light. One in a box, one in full light, one in partial light. Every day for a week, measure the height of each seedling and record the results in a table. You must record the height of the individual seedlings on each day. Once you have that data, you may calculate the mean of the seedlings each day and compare the mean height in the 3 different locations. Plot graphs of height against time. So, how are we measuring the seedlings? Get a ruler, hold it up against the seedling, and you literally measure in millimeters how high up the ruler it goes. The reason I've shown you this, because this is actually a drawn diagram, which you are going to have to do. AQA want you to be able to not only collect data and analyze it and talk about the data in terms of mean heights, but also some drawings of what you see. There are lots of investigations you could do with seeds and phototropism and growth, and you can't get everything just from the height. So, for instance, the direction of growth, like this one on the end, is kind of a bit wonky. This could be drawn, this could be represented, the observations could be represented with a drawing, and something like this would be good enough. Notice that it is labelled. Okay, last lesson we saw that and this was the explain task from last lesson. This is the table that you are going to need to make before you can collect the data. So you've got space for the days 1 to 7 as it's a full week, and the height of the seedling in full sunlight. So that's just for one of the three in millimeters. First seedling would have value, second seedling, third seedling, fourth, etc. And then there's space at the end for the mean being the average. There must be 3 of these tables, 1 for each condition: light, no light, and partial light. Right, so you are going to have to draw some diagrams based on what you see. Unfortunately, I can't show you. There's a video that I'm going to link and show my homework. However, you you're not going to be able to see one right now as you're at home. So I'd like to show you a little diagram, and I'm pretty proud of these. What you might find. So in the dark, in the light, and partial light, you've got this sort of growth. So first of all, let's have a look at in the light. Perfectly healthy seeds, as you'd normally expect. They grow up towards the light, provided the light is above them, and you've got nice, fat, juicy, healthy plants. Partial light, there is also directional growth towards the light, going upwards. However, you might find that there is just simply less growth than in the seedling in full light. You'd probably be able to tell me already, if you've already done the plants 1 topic about photosynthesis and the reason why it hasn't grown as well. But again, very, very similar, except for the fact it might just not have grown as strong. In the dark is pretty interesting because you'll probably find that there isn't any directionality to the growth. As soon as the plant germinates, it will just keep growing in a straight line, randomly, depending on how it's popped out of the soil. The growth will also be much shorter than the other two because it is missing that light for photosynthesis. So, quick recap. In the dark, grows in random directions and has weak growth. In the light, full light, it grows towards the light and is very healthy. And in partial light, it grows towards the light as well. However, it is better than no light growth, but not quite as healthy as the full light. Right. Pause the video now. I'd like you to match the type of growth to the light level. Okay, so you've got light, dark, partial light. Well, in the light, you will see strong directional growth. Strong, healthy in the direction of the light. In the dark, there is weak growth, and it's random directions all over the place. And finally, partial light is directional growth, just like in the full light, but it isn't quite as healthy as in the full light. What is going on here? Well, phototropism is the type of tropism that affects the direction of growth depending on the light, just like with this weird, gross broccoli that's growing towards the light. It isn't bending, but rather is growing towards the light. It is the response to the stimuli that the plant is getting, and the plants are actually experiencing light, and therefore it's growing towards that. The two that are in the light show positive phototropism. They grow towards the light. This is for photosynthesis, and this will hopefully explain what we see. Why do plants grow towards the light? This is a bit of a recap from the previous topics. Photosynthesis is a fundamental chemical reaction to all biology. It's probably one of the most important chemical reactions in biology. It's why I get so excited about it. Photosynthesis is a chemical reaction. It is chemistry that all plants do to make sugar. The point is to make sugars. As it's a chemical reaction, it can be written like this, with the reactants on the left and the products on the right. Water plus carbon dioxide makes glucose and oxygen. This is a endothermic reaction. It requires energy, and that's why I've drawn this lightning bolt to represent that. Energy or light energy is not a chemical, which is why I haven't written it down as part of the equation, but it still is required for it to happen. Again, this is the word equation, and down here is the symbol equation for photosynthesis. Right. Why are plants doing all of this? Yes, to make sugar, glucose being the sugar that it's producing. But also from the last lesson of the plants one topic, there are five. This is just revision. You don't need to memorize this for now, but it's explaining why plants grow towards the light. Plants use glucose for respiration. This is the chemical reaction to give the plants energy. They can store it as starch or fats and oils, which is kind of a store of this sugar to be used later on. Very importantly, though, plants use glucose to make cellulose, which is for building cell walls in every cell of a plant cell, every cell of a plant, and also to make proteins, which obviously are very important for transport, etc., inside plants. So without sugars, the plant does not get energy, can't store anything for later, can't build itself. That is why plants in the dark do not grow as well. How does the hormones come into this? Well, auxins are one of the hormones used for plant growth. It's made in the tips of the stems and the roots. In the shoots, concentration promotes growth. So in the shoots, if there is auxin, it increases growth. This is kind of the more complicated bit we've learned about. We spent a few lessons on this really. This is how the plants grow towards the light. Sunlight causes auxins to diffuse away. So this means that the shoot tip in direct sunlight has the lowest auxin concentration, and the shaded area has the highest auxin concentration. More growth occurs in the shaded part. This means that there is lopsided growth bent towards the light. The side in the dark grows faster. Those cells are more elongated. That means they've become longer, which pushes the plant towards the light. Right. We've been told that by AQA that we need to be able to record the results of seed length measurements, create a mean, and create labelled biological drawings to illustrate the effect of light or gravity. The describe section for today's lesson is based on the drawings, and then the explain is based on the data collection and what we do with the data. So again, we've got a table that looks like this. You need to be able to calculate the mean. To calculate the mean, you must add up all of the numbers and then divide them by how many you have. I expect all of us to be able to do this by now in Triple Scientist Year 10. However, a quick example would be if the numbers were 1 millimeter, 2 millimeters, and 3 millimeters, you do 1 plus 2 plus 3, which is 6. 6 divided by the total, divided by 3, which is 2. So the mean is 2 of 1, 2, and 3. You're not only going to have 3 numbers, instead you're going to have 8 numbers that I'm going to give you now. These, and I would like you now to be able to have a go at working out the means for all of these. So the explain task for today is be able to calculate the mean and draw it in a graph. There's not going to be space on the PowerPoint for later, so I'd like you to do the calculations now. Either you can fill in the data of your tables from last lesson, or you could just write down the means for day 1 to 7, please. Okay, calculate these. You will need a calculator in every single exam, but hopefully you've got access to a computer, so all of you have access there. Okay, good luck. Pause the video. When you come back, I'll show you the answers. Here are the answers. Notice that only one of them is a round number, full number. The rest are decimals. Okay, so once you have got 3 tables full of data and you've got average heights for each day, you can plot them onto a graph. What I've done is actually superimposed 3 graphs on top of each other, and you have got the height of the plant up the side, the time in days down the bottom, and the lines represent the growth. So you can see the blue, which is in light, grows the fastest and also ends up with the highest height at the end of the week. Partial light, again, not quite as high, but also still tall, and then in the darkness that there is growth, definitely is growth, because there are sugars stored in the seed. However, it is not quite as high as the other two. Notice that in my graph, I've got the days at the bottom, height up the side in millimeters, and a title for my graph, which is a graph to show how height of seedlings change with time for seedlings in light, dark, and partial light. You are only going to be given the data for light, and therefore you're only going to be able to draw the graph for one of these. And I am going to ask you to draw a graph in a minute. Okay, why do we see what we see? It is not easy to be able to explain what is going on, but this is kind of the point of science, to take some observations and explain using the models that we understand. So, in the dark, you've got random directions weak growth with a mean height of 3.8. This is just data that I've collected. When we do it in the lab, we might have different ones. But notice that it is the smallest out of the three. Light is the tallest out of the three, with 8 millimeters at the end of the week. And partial light is 6.8, which is, again, in between those two. So again, this is the part of the lesson where I'd ask you to turn to your neighbour and have a discussion of this. But unfortunately, we're not in the lab, so I'm going to ask you to go and talk to somebody else. Anybody that you can find, grab them, stop them from doing whatever they're doing, and say, "This is really important for my science homework. Can you tell them why did you find what we found using phototropism? Try and explain it using hormones and phototropism." Pause the video. We'll go through the answers when you come back. Right. How can we explain our findings using phototropism and hormones? The two batches of seedlings that were exposed to light or partial light grew towards the light and showed positive phototropism. This means they grew towards the light. What's going on here? This is not easy. The auxin in the tips of the shoots diffused away from the side closest to the light, stimulating growth on the opposite side. These cells elongated and turned the direction of growth towards the light. The seedlings in darkness did not show phototropism and grew in random directions. The auxin was evenly distributed in these shoots, so they grew in straight lines in whatever direction they were already pointing in after germination. So this can only explain the direction. Phototropism is to do with direction of growth, and therefore this is what I've explained here using our knowledge of hormones. Right, now it's come to the point of the lesson where we're going to do some writing. I'd like you to, underneath the driving question, if you haven't got that, make sure that it is there. And for the describe, explain, use, and create, have a go at answering those. You're going to need to draw and label diagrams of the seedlings. We've seen those already. I'd like you to draw them yourself. We've calculated the mean already, but I'd like you to have a go at drawing the graph of your results. So this is another view of the questions. Draw a graph of these results. This is for the light. You've got days up to 7, and these are your mean heights that we calculated before. You can do this on graph paper if you've got any available to you. You could do this on Microsoft Excel. This is how I did my graphs. It's up to you how you draw the graphs. However, you're going to need to be able to draw them using rulers and pencils. Okay, we've had plenty of practice already. I'd like you to have another go of drawing a graph today. That's for your explain task. The use section is to explain the results using phototropism, so what we've just discussed then, and have a go at just explaining the results using photosynthesis as well. So these two could just be a couple of paragraphs. Give yourselves about 10 minutes or so to do this. Perhaps the graph might take a bit longer, 10, 15, 20 minutes on this. We'll go through the answers when you come back again, when you unpause the video. Okay, thank you. See you in a minute. Okay, time to answer some questions. Time to get your green pen ready. If you have not got a green pen, then that's okay, just any other colour. The important thing is that you are giving yourself ticks, but definitely correcting your work if you're missing anything. So diagrams could look simply like this, thin lines, but the important thing is that you've got directionality here. You need to be able to compare the three, and therefore this would not be good enough. You're going to have three of them in the dark, in the light, in the partial light. I need you to have labelled these. Perhaps you could have said what you see, but the important thing is that you've got labels. So, for instance, if you've drawn the Petri dish, you've labelled the Petri dish, etc. Okay. The second bit is your graphs. Hopefully your graph could look something like this. On the first day, you've got the lowest height. The last day, you've got the highest height. This is what you would end up looking, this relationship here. In the exams, I would have to go through all of your answers and make sure that you are within, I think it's half a square even. You have to make sure that they're close, but I'm not going to be able to mark those. The important things as well is that you've got a title for your graph. You've also got a axes title, so time is in days along the bottom, height is in millimeters up the side. Okay, it should look something like this. And our explanations, we've been through this already, but how to explain what we see using phototropism and hormones. The two batches of seedlings that were exposed to light or partial light grew towards the light. This showed positive phototropism. The auxin in the tips of the shoots diffused away from the sides closest to the light, stimulating growth on the other side. These cells elongated and turned the direction of growth towards the light. The seedlings in the dark, however, did not show phototropism and grew in random directions. This is because the auxin was evenly distributed in these shoots, and so they grew in straight lines in whatever direction they were already pointing after germination. Right. A little bit more on top of that. So the final one is explain what you see with phototropism and photosynthesis. I've only written the photosynthesis bit as it kind of complements what we've just written just then. So the seedlings in the light grew most of all as they had access to more sunlight. This meant that they could photosynthesize more than the others. Photosynthesis produces glucose as a product, which is used by plants for respiration and building. Seedlings in partial light grew well, but were limited by how much light they received. Those seedlings in the dark could only grow using the sugars that were already stored in the seed. When those ran out, the plant could produce no more. These seedlings were stunted. For extra points, have you written down what the chemical equation is for photosynthesis? It'd be water, carbon dioxide, makes glucose and oxygen. Okay, fantastic. Not easy, this required practical. Loads and loads and loads of information, and I promise it will be easier once you've had a go of it yourself to memorise. You need to write your Today I've Achieved sentence now. Please pause the video, write Today I've Achieved, in the gap, write describe, explain, use, or create, depending on what you have achieved, and say why, because I could explain the results using phototropism, etc. Pause the video, write that. Okay, and then finally, please do not forget that you have a 10 questions show my homework quiz. This will not only show us that you can remember some of the facts from today's lesson, but also it will help consolidate the information that you have learned today by it's basically going to be revision, isn't it? Thank you so much for your concentration over the last two lessons. Required practical, not easy. We'll be looking at something slightly different again next lesson. Thank you very much for your time. Bye for now, Year 10.